Shall we go with our next speaker?
I think so. All right. Good afternoon. My name is Kalen Rasmussen. And I'm a recent graduate here at School of Mines. Graduated with a bachelor's degree in chemical and biochemical engineering. In a few months, I'm going to leave for Germany. I will participate in a Fulbright fellowship with Andreas Kepler at the University of Tübingen. I've worked with John for some time now, and I've worked on a few projects ranging from the astrobiology of a high Arctic glacier, and ranging from that to the microbial diversity of sour ciders. And no matter how important I think my role in the lab is, realistically, I know that I'm just kind of that annoying little brother who just keeps asking too many questions to everybody else. But as you all know, the real reason I'm here is to introduce John Spear. The more and more I find out about John, the more and more I am kind of enthralled with just kind of his life and where he's gone from, the time that he's been around. John started, he took his dear sweet time getting all the way through school. Started out at UC San Diego. Got his undergrad there. Then he got his master's and PhD here at Colorado School of Mines. And then he did a postdoc stint at Norm up at UC Boulder. And the thing about John is his exploits go far and beyond his academic career. I recently found out that for some time now that John, he worked at ski resorts as what I would like to call a professional ski bum. He became very, very proficient at all three types of skiing, telemark skiing, alpine skiing, and snowboarding, which to me is just amazing because I'm not even good at one of those things. So that's really cool. And his love for the outdoors expands even beyond that. And it's pretty evident in the fact that he was a Knowles instructor for some time, and he used the great outdoors as his classroom and his way to educate others. I think knowing those things, it's no surprise to me that he is where he is now. He's here teaching at School of Mines as well as doing some pretty cool research in some of the harshest and most isolated environments in the world. Recently, John went up to the high Arctic on Ellesmere Island, which is one of the northernmost landmasses in the world. And he did some really cool research up there, but I mean, secretly we all know that the reason why he has to go up there is because he just keeps getting into trouble down here at Yellowstone. And I haven't known John for that long, but he does like to pass on a lot of useless and even more useful tidbits of information. And one time I was sitting down with John and just kind of talking about some stuff, and he was talking about when he was up in Ellesmere Island, and he was like, you know what? I gotta tell you something, Caitlin. One of the most useful things up there is snow. And I was like, all right, that's cool. That's cool. And he kind of went on with that, and he really explained to me the so useful ways that you can use snow when you're in the tundra. There's no trees that produce leaves, and for that matter, you're a little bit low on TP. But so, with no further ado, John.
So as far as snow, snow is nature's finest butt wipe. And if I had my way, every toilet in the world would have a little crushed ice machine next to it, so you can make a little snowball. So if you're around the wild side, snow gets the highly recommended. This is a place where we use that, we did that, it is what it is. And this is a project that's funded by NASA Exobiology. And we were up here a year ago, and we want to talk about this. This is up on top of Northern Eldorado, but we'll get there. First, I want to start with several thanks. Thanks go to our research team on this project. It's Steve Crosby from Natural Resources Canada. He's in the red jacket right here. And then Alexis Templeton, standing next to him from CU Boulder, her graduate student Graham Lau in the hat, and Chris Trivedi, who we introduced this morning, is sitting right there. So the five of us were up there for about 3 weeks last year. It's the wildest, most remote, most natural ecosystem I've ever been in, so it's a pleasure to be there. Polar Continental Shelf Program Canada supported us in this whole role. It takes a long time to fly it there. It costs about $20,000 a person to land a person there at this place. It's 4 or 5 plane rides plus a helicopter ride. I want to begin a little bit of shameless self-promotion in that I'm co-director of the International Geobiology course with Frank Corsetti at the University of Southern California. Frank and I have been associated with the course for about a dozen years. We've been co-directing it now. We're going into our 6th time in 2 weeks. Next year will be our last year of doing this, but if you know of mid-level PhD students who are interested in anything that has to do with the rock microbe interface, we would love to have them take the course. It's an amazing thing, and what it allows us to do is think about, I think some of the best science in the world, as we've heard some today, is happening in interfaces. So here it's microbiology and geology coming together. It's kind of glued together by geochemistry. And we look at this from the basic side of things because microbes don't leave fossils, right? But rocks do. And so to be able to correlate with stable isotopes, clumped isotopes, thorium, uranium dating, you can start to get into deep time with rocks, and then you can start to think about, well, was that a stable isotope signature reflective of microbial processes at that point in the rock? This is particularly true for things like stromatolites. So we're thinking about that in a basic sort of way. And we also think about applied problems in geobiology. And one of them is like Mitch's sewage pipe. So I look at that pipe and I say it's made out of metal. Metal wants to do one thing. All metal wants to return to the ore. It wants to go back to where it came from because it's not a thermodynamically stable state. And who makes it get there is abiotic corrosion, which is chemical corrosion, and then there's biotic corrosion. So a lot of what we do in our lab is microbial influence corrosion of steel, for example, in like Mitch's pipe. And so I see those as both aspects of geobiology. And those are some of the things that we study. And I'm only going to talk about one story here today, and it's sulfur. And sulfur, just some basic things. It's the 10th most abundant element in the universe, 16th most abundant here on the Earth's crust, 6th most abundant in the oceans, 3% of the Earth's mass is sulfur. 25 isotopes, which is a lot. It has at least 30 allotropes, like graphite and diamond for carbon. There are many forms of sulfur. As a caver, as Norm would attest to, you go into caves, there are different kinds of sulfur doing different kinds of things, making different kinds of minerals. And then you've got this wild 8-electron transfer oxidation state of redox from H2S to sulfate. Global sulfur cycle is pretty much throughout our entire planet, everywhere. Not so much indicated on here as deep subsurface cycling, but it's certainly in Ana Luisa's vents, obviously. But you have volcanic emissions, you have sea foaming and sea sprays. You have the burning of fossil fuels, which is putting up lots of SOx, which is contributing to air pollution, for example. And then all these things have various rings associated with them. This is simplified. Many of those rings have microbes involved in them. I think I already said that for the most part. So here in Colorado, there are a lot of pyritic ores, high sulfur ores, and when you expose them to oxygen and bacteria, you start getting acid rock drainage or acid mine drainage. And those are all things because the sulfides are binding metals. And this is a big problem in the Rocky Mountain region or anywhere where there's mines. Acid rock drainage occurs normally. It's a sulfur-driven process, but it also happens when we make these mines. And those are things to think about. The biogeochemistry is, I think, obviously it's a critical element for amino acids and for proteins. And so you think about how life is built upon a foundation of carbon and sulfur. And it's dominated on the Earth's surface. The ones that are studied are like, when I was a grad student, I was working on desulfovibrio desulfuricans, as well as desulfonobactam, because they both not only metabolize sulfur, but they metabolize uranium. So you can take uranium-6 to uranium-4 as a treatment process, for example. So this project is funded by NASA. I'm trying to get you into why it's I want to think a little bit about space. And so if you look at Jupiter, it has 67 moons. And it has a bunch of active volcanoes on Io, which is, it's north. Io and Europa are its two most famous moons, I would argue. Ganymede's right up there. And then Europa's got this water and ice environment and tenuous oxygen being slipped out by something going on with water. Europa specifically is crisscrossed by all these cracks, crevices, lines, and creases. And we want to try to figure out why that's there with an upcoming mission that NASA wants to do in the early 20s. So it's not far away from us for now, right? And so if you want to stick something up there, how do you determine if there's the possibility of life? And how do you determine the possibility of elemental proportionation? to see if he can aid in the determination of is there life there. And so one of the best, or probably the best analog for Europa on Earth is this spring. And this spring right here is on ice, and you get this color coming out. This is Steve Grasby ice climbing on a sulfidic spring at Borg Fjord. And it's probably the best analog for Europa in terms of the element sulfur. The best analog for Europa in terms of iron is Blood Falls, Antarctica, probably. So one's in the south, one's in the north, but we're interested in both these things. Steve first saw this in a helicopter flyby in 1988. So he saw ice and saw sulfur, and he said, what is that, right? I mean, why would you have that happening down there? With a couple of surveys after that, the first field research was in 1999, and then we've had several field sessions since, including last year in 2014. Steve is going back this year for, I'd love to know why it does that. Steve is going back this year for probably a day and a half in 2015. So where it is, here's Ellesmere Island, Canada. This is Greenland right here. This is Ellesmere Island. I'm just zooming in for you. This is Borg Fjord right here. Zooming in a little further, this is our research site. This is the Borg Fjord Pass Glacier right there. And we're going to be studying this spring, which is on the toe of that glacier. This is zooming in further with a Landsat view. And this is a little model right here of where the spring is. These black lines right here are faults. So down valley, there are faults heading down here towards the ocean. And these crisscross the whole valley, and they're very pronounced. And what's important about the faulting is that it's making the subsurface a little more available for glacial meltwater. We'll talk about that here in a second. This is what it looks like from the air in 2006, a trip that I was not on. And let me say that Alexis Templeton and I, professor at CU Boulder, we both started our faculty jobs in 2005. We both knew Bob Pappalardo, who was formerly in LASP, the Laboratory for Atmospheric and Space Physics at CU Boulder, who's now at JPL. And Alexis and Steve and I, we kind of pooled some startup monies from our different entities to start thinking about this site. So though my first time was there in 2014, Alexis and I have now been thinking about this site for about 10 years. And so the first time to see it was great this year, but it was good to go through and look at the old pictures from 2006 right here. The sulfidic spring is up here on the ice, and it has these icings that flow all the way down towards the ocean. And you can see that's looking down the valley. The fjord is right down there. From the toe of this ice right here down to the ocean is about 20 miles or thereabouts. And you can't just walk around it. It's difficult walking on tundra. That's what it looks like. And then the spring moves around with time. Because you have this huge glacier that has different plumbing, right? The cracks, crevices, the plumbing is changing, as well as the subsurface flow is changing. So in 1990, the spring was here. 2000, 2001, 2007, it's over here. This year it was about right here. And if you look at it 1 year, this is Steve standing next to it 1 year, here it is the next year. Because the ice is moving, melting, changing, right? All this sulfur crust right here becomes a pile. And this pile, you can say, well, what are the microbes? Are there microbes responsible for the formation or entrapment or precipitation of this crust? And right here, this is what it looks like a year later. Can any of this serve as a biosignature? It's one of the things you could think about, right? If you had this happening on Europa, how could I use your tricorder to look at that and say, boom, there's life? It would be nice. The tricorder still doesn't exist, though. This is just Catherine. Catherine Wright was working up here. This was her spring, was way up on the ice. This year we were over here. And you get this flow of sulfur coming down, going down the valley with different kinds of communities associated with it. But in 2011, that's her spring. Pretty much disappeared, right? So it's a very transient thing. Glaciers are very dynamic environments, just like hot springs. Chris Trinetti took this picture looking north. This is this year. This is one place we call the blister zone right here. It's a little bit overcast light right here, but I'll show you some other pictures. You can see where the yellow kind of is down here on the toe of this glacier. And Catherine Wright's spring was right up over here several years ago, and now it's down here. That's where you get, this is from looking at a helicopter down at our site this year. This is like Sulphur Falls right there. Just beautiful yellow ice, right? And quite a unique site. When you go here, it's very sulfidic. It does smell like sulfide. And you can see the effect of what things are happening where. We have these proglacial icings that form down there, and they trap and bind or emit either sulfides or sulfates that are being transported down valley through the water, or there's even fizz coming up, right? The subsurface is fizzing below this glacier because of its mass. This is our research site right there. So we took a time-lapse camera with us. And one thing I like, I'll just show you. So this is the Arctic. A lot of people wonder what life is like in the Arctic, and so we're going to buzz through this relatively quickly. So that's what the site looks like. This is probably morning, sunny, like maybe 10 a.m. And then you're having time goes by as this changes. I'm going to speed this up here to about the middle just so we don't watch the whole thing. So this right now, that's about midnight. And it's nice because you can watch the sun do its thing in the north. It never sets, right? And so as you go along, we're all probably sleeping. We stuck to a day schedule of working. It's like I learned this from caving. When can you go? One of the questions of caving is, can I go caving at night? Sure, it's always dark in a cave, right? And you can work anytime you want because it's 24 hours of light. But for most of us, watch how the rocks are sliding here because the sun's hitting it. For most of us, we're acclimatized to working during the day, so we stuck to a day schedule while we were up there. You get used to sleeping in the light. This is probably about 3 a.m. And it's, when you watch this, it seems like it looks the same every day. On time lapse, it's not, right? Because this is actually all ice underneath this rock. And so you have these things that are the size of a house that just goes sliding right along, right? And then you can watch us come to work. This big waterfall formed while we were there, coming off the Sulphur Falls. What's that?
It was a late start that day.
A late start that day. It takes a while to get around. What's that, Tim? Right? That's it. Time-lapse cameras can be good for field work. So this is a proposed model for how we think the spring is working. I'll show you a refined version here pretty quick. But you have these mountains on either side. You have glaciers, and then you've got pressure. You've got glacial melt coming down, and it's going through different kinds of rock and different kinds of subsurface. Then you have fault lines right here, which are striking and slipping, and that's changing the geochemistry of what's possible because you're altering the water, rock, and reactions in the deep, as well as in the surface. You pick up sulfur and you transport it to the top and you bubble up a sulfide spring. Steve Grasby has been here before when it's just a straight tube of yellow water shooting out of the ice, like brilliant yellow. We didn't get to see that this year. For us this year, it was like probably that tube of yellow water was beneath a sponge, and all the yellow is seeping out through all the holes of the sponge is the analogy that I would use. So you're focused on, is this microbial oxidation of sulfide to elemental sulfur? Is it abiotic? Is it biotic? This is a model that Chris Trevetti is proposing based on what we saw this year. You got glacial outflow up here. We had this ice blister, which was intriguing because we think it was a cap that kind of formed on the spring. It was a very sulfur-rich cap. And then there's permafrost down here that's getting in the way. You could have deep subsurface microbial activity that's altering your sulfur. And because what you're thinking about is thermochemical sulfate reduction. If you're going to do that, it takes temperatures of at least 140 degrees C, which are probably unlikely underneath a glacier, at least in the near depth underneath the glacier, right? And then there's biological sulfate reduction, which could be happening down in the deep, and then it's just coming up as the sulfide, it gets reprecipitated as sulfur. So the questions that we're specifically trying to answer are a couple. What are the physical chemical factors that allow for the deposition of the sulfur on the ice, right? How are the microorganisms involved in this process? What can the environment tell us about other systems like Europa? What can we infer from here to other places that are astrobiologically relevant? And so to do this, we start with the who's there, which we've heard some today, using MiSeq and HiSeq for 16S and 18S ribosomal RNA. But what's good about our system now is these are limited communities I'll show you in minutes that are ideal for metagenomic probing. And so we want to understand the metagenome of that. And then working with Alexis has a postdoc up in her lab who has many cells in culture. that came from this site. So what we're going to do is play a transcriptome under different kinds of conditions and see what that kind of story tells, and then get into further gene activity experiments and see how active this is. So the idea is this will be Chris Trivedi's thesis, and we're just starting into it now. This is not going to be a data-heavy talk because he's just getting going. But here's some geochemistry about how the spring fluids are changing and different versus surface. So you've got fluid chemistry on meltwaters, and you have fluid chemistry on deep waters. And then you wonder, how are these things mixing? So you've got sodium bicarbonate versus chloride and calcium. It's calcium rich. And you can use things like that to say, well, that water is coming from a deep subsurface versus a meltwater on top. And so we suggest that the meltwater is the source for the water that goes down, probably diffuses and melts or dissolves or microbially reduces sulfate in the subsurface and brings it back up as sulfide. This is what the top of the ice looks like. It gets this sulfur crust on it. It's brilliant yellow. You can put your nose down to it. It's a little sulfidic. And it has this kind of strange consistency, which you'll see here in a second. This is a little pool of what sulfide looks like when it's fizzing up through the water. You get these sulfur caps on it right here. If you bubble strip this water, so we'd stick a tube in there and look for dissolved gases like dissolved aqueous hydrogen. It's 29 molar hydrogen, which is a lot of hydrogen. In Yellowstone National Park Hot Springs, when I was working with Norm, we measured hydrogen up to 300, 400 nanomolar, which is good for a terrestrial system. It's not millimolar like a hydrothermal vent zone. But 29 nanomolar is certainly 6 or 7 times more than you need to be a microbe living in there. So they've got sulfide, they've got hydrogen, they've got water, and they're making a living potentially. It has this nice crust that forms on it. We scoop that up, and then that's what we want to do genomic work on. But we're finding out that there's not a whole lot of DNA biomass associated with that, but there's probably enough to work with. Right, Chris?
Yeah.
Yeah. And so some primary sequence that's back right now, just looking at studies. So we have a fair amount of epsilon proteobacteria and gamma proteobacteria, both of which Julie up in Alexis Templeton's lab has in cultivation, and they represent the dominant biomass in a couple of these sites. So that's why we want to target a transcriptome on that under different sulfur conditions, under laboratory conditions. Thought about doing a transcriptome under field conditions, but that becomes a harder thing to do. So we're going to do it more under kind of a controlled condition. If we zoom in and think about just the file of a particular single site, thinking about proteobacteria being the dominant part of it, this other 2% is what's interesting, because at least Julie, she's got one thing cultivated that's coming from the firmicute, and it's only comprising one small part of that with this community that's dominated by proteobacteria. So we're wondering, what are these 2% players doing in terms of sulfur metabolism? Maybe this is doing all of it, but what's that 2% doing? It's something that we want to consider. So that was my data-rich talk, telling my story of my place. And then I wanted to shift in to say a few things about Norm that hasn't come up so much today, but it's this, in that I've had some amazing, wonderful experiences with Norm in the field. Norm's a research scientist who does amazing lab work, but he also loves field work. I've been to Yellowstone, Guerrero Negro. I was in a cave in Glenwood Springs, Colorado with Norm, and it was a hot, steamy vent thing that's a dark hole in the ground, and I was the first person to go in. I had my light on, and then I disappeared into the dark, and Norm just pokes his head over the hole, and he goes, "Hey, John, are you dead?" And he goes, "Are you dead yet?" And then he said something like, "Shit, I'm not going in there." It was great. But this is up in Yellowstone with Jeff Walker, sitting right there. Jeff and Norm checking out a site. To understand and be in the field, Norm would always say, if you want to understand something, you have to gather your own DNA. I think that's very true. You got to get your own sample, because then you understand the context of where it came from. And this is something that we do. This is Tom McCollum from CU Boulder, and Norm, this is at Washburn Spring. If you sit right there, or right here, it's the same spot, the soil is dry unless it just rained. If you sit there when it rains, pH of the soil is around 0.5 or 1, so you're pretty much just sitting in sulfuric acid. So my grad student Jason sat there while measuring the pH one day and stood up. Not only was the butt on his pants gone, So was the butt on his underwear. And so I had to loan him a pair of pants to walk out. I think these two guys right here are marveling at the fact that there's a dead bison in the pool right here, and that does happen in Yellowstone. This is Norm gathering a sample in the Norris Geyser Basin. We were talking about the spongy, foamy stuff that the microbes are filling on here, and it looked a lot like activated sludge, right? And so Norm was pondering on, what's that stuff doing? Which we then take back to the car so he can do this, because you have to have a field microscope, and you do this in the parking lot. And that's Norm and the microscope in the parking lot of Norris Geyser Basin. And then I just wanted to end there and say, doing things like this and doing things like we talked about today, it's just a wonderful thing. So does anybody have any questions on Ellesmere?
About the rapid water flow.
The rapid water flow. Yeah. I got a minute. So the first day we were there, where the blister is, Steve Grasby was up above us, and he was taking pictures of us working at a lake that was right there. And he came back. So he came back down, and when he got back down to us, he was soaking wet. And he was soaking wet because he fell into a pool up there above us. And he was getting cold, and it was cold out. And so we walked away from the lake to get kind of out into the sun. And then Alexis and I heard this sound that sounded like a jet engine. And then Chris and Graham and Steve all heard it. And it wasn't, you think, is that a low-flying military plane, or like what's going on? It wasn't that, it was a flash flood. It was a glacial flash flood that was coming down the glacier. And it deposited 20 feet of snow, 30, 50 feet wide, 20 feet deep, to the place where we had just been standing 1 minute before. And so you think you're very careful in the field doing your thing, and then something like that happens. It can be a sobering event, right? But it's the dynamic nature of glaciers, and it's an interesting thing to see and be part of. So watch what you do when you're in the field. Always be aware of your surroundings is my answer to that. Right? Any other question? Okay, that's all I had. So I'm going to stop there, and then I'm going to say to adjourn the meeting. But to do that, I want to say thank you all for coming. I want to thank the students, introducees who introduced all the speakers, and I want to thank all the speakers who were here. So for me, 24 hours of a great thing. The science talked about last night and the science talked about today is remarkable. And I think that this is a great sized meeting to share ideas, communicate, and talk about something that is the wonderful achievements that have come out of Norm's lab. Just the direction that we have all gone into these different areas. Mine is rock and microbe, and Tom's is doing ecology, and Mitch has got all the stuff, and Gary's got evolution. It's remarkable, and that's just an amazing legacy. And I think we can all say thanks. So let's say thanks to Norm one more time for making all this possible.